Today we will be dealing with the topic of bioenergetics. Bioenergetics is the basis for photosynthesis and respiration. Like when we take life forms, when we take all biological processes in all life forms, whether it is bacteria or plants or microbes, we understand that predominantly the solar energy is fixed into plants and animals. So when we are talking in terms of energy, the basic source of energy for all life processes is the sun. We get energy from the sun. So all life processes or all biomolecules as well as cells convert the solar energy for running the different sort of a life process. Now predominantly when you take the sun is rich in hydrogen, and by both fusion and fission there is release of helium. Parallel to release of helium, there is an enormous amount of energy which is liberated. This energy which is liberated drives all the biomolecules and cells to perform their life process. Among the basic life process we have is photosynthesis, and we have respiration. In plants, microbes as well as in archaea, we have photosynthesis and respiration. Now when you take the processes of photosynthesis and respiration, they are predominantly carbon based. So carbon is the most important molecule in the biosphere. So all life forms on earth are carbon based. All life forms and their life processes dependent on photosynthesis and respiration are driven by carbon. Most of the biochemical reactions which occur in plant cells are oxidations. The objective of any particular biochemical reaction is oxidation, and by oxidation we have the formation of energy, and energy in turn in terms of ATP, that is the energy currency of the cell, is the basis for biochemical reactions. Thereby the reason why we have to link up physiological processes like photosynthesis and respiration to energetics is because in chemical reactions as well as with relation to different sort of physiological functions, always energetics forms the basis, because by energetics, that is by energy transformations, we convert from disorder to order. That is, from the free energy state or from entropy, there is an ordered state, and in the process energy which is released, the energy which is released is helpful for driving different sort of biochemical reactions. Thereby we say that all life is energy transforming systems. Most of the life forms are carbon based, and again the major reactions are oxidants, and energetics is the basis for all the sort of life processes. Now, in bioenergetics, we are dealing with photosynthesis. When you take photosynthesis, there are plants, microbes and archaea which are rich in chlorophylls. Now, because they are rich in chlorophylls, in the presence of a chlorophyll pigment and in the presence of energy, that is solar energy, they can drive reactions all by themselves. Photoautotrophy, we have it. That is, they are able to manufacture their own food materials in terms of glucose by using the light energy, and it is driven by the chlorophyll pigments which are present in them. So, such organisms are termed as photoautotrophs. Normal examples of photoautotrophs are plants, we have microbes, as well as the most primitive of the life forms called archaea. We also have cyanobacteria, blue-green algae. All of them are termed as photoautotrophs because they have got the sort of a chloroplast. Now, in addition to this, there are some other organisms which can convert inorganic substances into complex organic substances. So, these are also termed as chemoheterotrophs. Photoautotrophs can take inorganic energy or inorganic matter, and in the presence of sunlight, they can manufacture complex food substances. That is, from simplicity to complexity. This is the attribute of photoautotrophs. When you take the photoautotrophs, whereas when you take the chemoheterotrophs use organic substances like those present in the soil, and to this they add the energy from the sunlight, and thereby they release certain compounds or chemicals which are predominantly made up of elements. So, organic substances getting broken down to simpler substances is an attribute of chemoheterotrophs. They are unable to manufacture their own food materials because they do not have chlorophylls. Now, either way, whether it is photoautotrophs or chemoheterotrophs, the basic principle is when you take photosynthesis, life on earth directly or indirectly is powered by photosynthesis. Is powered by photosynthesis from the sun. Thereby, all life is dependent on photosynthesis. All life is dependent on photosynthesis. So, the entire driver in terms of bioenergetics is the process of photosynthesis. Now, when you take photosynthesis, during photosynthesis, what exactly happens? It is a process, it is a physiological process, it is also a biochemical process whereby there is energy capture. A. There is energy capture. Energy capture occurs by excitation of the chlorophyll molecules. Then B. There is energy stabilization. So energy stabilization occurs due to the presence of, it occurs in the Kelvin cycle. When you take the dark reactions, energy capture relates to the light reactions, energy stabilization relates to the dark reactions. Then again we have C, energy yield. So the energy yield process relates to the formation of ATP. So once we have energy capture, energy stabilizing and energy yield process which occurs in green plants, is predominantly termed as photosynthesis. So photosynthesis is a biological process whereby there is light, dark reactions, energy capture in green plants, as well as it is commonly seen in the case of the autotrophs. Now during bioenergetics, there is always energy transformations. When you say energy transformations, it predominantly refers to the principle of free energy, entropy, chemical potential, oxidation reduction and chemiosmosis. So here when you talk about free energy transformations, we have to be looking in terms of what is the free energy of the molecule. We also need to look at the concept of entropy. How much of heat does a particular molecule generate? We need to think in terms of chemical potential, the capability of the particular molecule to move in view of its chemical structure. Most importantly, it is based on oxidation reduction reactions which are coupled. When you say coupled, the meaning of the term coupled is it is linked. So all bioenergetic reactions, whether it occurs in the photosynthesis or it occurs in respiration, whether it occurs in the chloroplast or in the mitochondria, they are all oxidation reduction reactions which are coupled. They are coupled with relation to B, chemiosmosis. That is the way how protons and electrons are generated across a sort of a membrane. So here this chemiosmosis is membrane bound and there is an uptake of H plus. So in all biochemical reactions, the basis of bioenergetics is there are oxidation reduction reactions. The oxidation reduction reactions are coupled, they are linked together. This in turn is linked to chemiosmosis, that is the transport of a particular sort of a chemical across a particular membrane, and during which there is a sort of a potential and it is membrane bound. We are looking at what is the uptake of H plus and also what is the uptake of protons. How do protons and electrons move across a membrane causing chemiosmosis, and how it is linked to oxidation and reduction. This is the basis for the study of bioenergetics of photosynthesis as well as mitochondria. So we understand when we are dealing with relation to photosynthesis, which is the process of combining carbon dioxide plus water in the presence of chlorophyll and light and primarily forming carbohydrates and liberation of oxygen. There is always ATP synthesis. Photosynthesis always has ATP synthesis, and during photosynthesis there is also ion exchange. across membranes. So, photosynthesis is a process wherein normally it is very important in terms of ATP, in terms of energy capture, which is also coupled with ion exchange across the membranes. So, this process wherein ATP is coupled with ion exchange is termed as energy transformation. So, energy transformation in living systems, when you say living systems, it means mitochondria, it could also mean chloroplast. These energy transformations are linked to the biological molecules, hence it comes under the purview of the term bioenergetics. So, primarily when you take photosynthesis is an example of energy transformation, is an example even of energy transduction. Transduction primarily means how one form of an energy is converted to another form of an energy, which is termed as energy transduction. When we look at energy transformations in living cells, like if you take a photosynthetic cell, in a photosynthetic cell always energy transformations that is, how does a solar energy get converted? Energy transformations are always coupled reactions. When you say energy transformations are coupled reactions, it means they are linked reactions. And primarily here, the free energy of ATP, once ATP gets formed, adenosine diphosphate plus an inorganic phosphate, we have an ADP and we have a terminal phosphate. This has got lot of energy and it results in the formation of ATP, wherein the terminal phosphate is high energy bound. This energy A of ATP is linked to, this energy is linked to coupled phosphate reactions. That is, whenever we have a reaction wherein we have A plus B forming C plus D, if it involves ATP, if ATP is synthesized over here, that is, we have adenosine diphosphate plus inorganic phosphate, which will couple and go to form ATP. So, these reactions are very, very important. So, these are termed as coupled reactions or linked reactions. So, always in energy transformations of linked reactions, or in the case of photosynthesis or in the case of respiration, we always have coupled phosphate reactions. When we take bacterial membranes, or else we are studying chloroplast membranes, or else mitochondria membranes. We are dealing with relation to membranes. We are looking at membrane, their biochemistry, that is what it is made up of. We are looking at their physiology. How is the membrane? Is it acidic or alkaline? What is the pH of the sap of the chloroplast or the sap of the mitochondria? and how is ATP synthesized. So it's very, very important that we have to be looking at these particular things, because when light falls, when solar energy or light energy in the form of a photon of light or in the form of a wave, when it falls on a chlorophyll bed, the chlorophyll bed, energy gets transduced. When you say energy is transduced, energy, that is when you have got solar energy, and solar energy comes in the form of a particle or as a sort of a wave, and it hits a chlorophyll bed, the chlorophylls pick up the sort of an electrons from here, and they in charge get excited. So when they get excited, electron moves through the chlorophyll bed upwards. So this method of transfer of a photon of light, when it hits a chlorophyll bed, when the chlorophylls get excited and they lose an electron, this process is termed as resonance transfer. So whenever we want energy transduction, there is a technique there is a mechanism which is termed as resonance transfer. That is, the chlorophyll molecules do not get separated, but they transduce. That is, just by touching, one chlorophyll touches the other chlorophyll, and through the chlorophyll bed, which is just like a mosaic of a sort of a chessboard, the electrons move through the sort of through the chlorophyll bed, just like in the form of a wave. So this sort of an energy occurs in nanoseconds and picoseconds, and the technique is termed as resonance transfer. So this forms the basis, or this transfer is coupled with chemiosmosis. So electron transfer is coupled with relation to flow of protons. So this is the basis. This forms the sort of a basis for study of any particular biochemical reactions. On one side we have an electron flow. Imagine we have the chloroplast membrane, we have the outer chloroplast membrane and the inner chloroplast membrane. Or take a sort of a thylakoid. In the thylakoid, primarily we have certain units which are termed as quantosomes. Now quantosomes are light harvesting complexes. Now when you take a light harvesting complex or an LHC consists of a cluster of chlorophyll beds. So in the cluster of chlorophyll beds, all the chlorophyll molecules will absorb the light energy and they will transfer it to a major reaction center, RC. So chlorophyll bed with RC along with other accessory pigments forms a photosynthetic unit, and the photosynthetic unit is termed as quantosome. So to quickly recapitulate, we have membranes. Membranes are involved in energy transduction. When you say energy transduction, we have solar energy. Solar energy hits a chlorophyll bed. The chlorophyll molecules get excited, and the transfer of electrons through the chlorophyll bed to one important reaction center, this is termed as resonance transfer. When we have the chlorophyll bed with the reaction center and the accessory pigments, the entire unit is termed as a quantosome. And when there is a quantosome, there is resonance electron transfer, and parallel to this, on the membranes itself, there is proton or chemiosmotic hypothesis. Now when you link these two, this constitutes the study of bioenergetics of photosynthesis and mitochondria. When we come to the sort of light reactions of photosynthesis, The light reactions of photosynthesis are also called as photochemical reactions. They occur on the membranes of the thylakoids. Now, you would be familiar with certain compounds like called as plastoquinone, plastocyanin, quantosome, cytochrome F, cytochrome B6 and others. Now, all of these particular compounds which are present in the membrane, each one of these particular compound can get simultaneously oxidized and reduced. So, we are saying that oxidation and reduction are coupled in photosynthesis. We can just study this in a particular manner. Like, you take an example. You think you have a molecule A. The molecule A is reduced. So, when you say molecule A is reduced, what do you understand? A molecule gets converted to B and B loses an electron, minus an electron. So, when A, which is reduced, gets converted to B, B gets oxidized. So, when A molecule is converted to B, from reduction it becomes oxidized, and in the process it loses an electron. You take the similar situation, you take B, which is oxidized, oxidation, and to this you add an electron. When you add an electron, again the compound B itself can get reduced. It is important that we understand this, because if you got a compound A, it undergoes reduction simultaneously along with another compound which undergoes oxidation. Thereby, A is oxidized and B is reduced. B is reduced. So, A is called a reductant and B is called an oxidant. In all the sort of photochemical reactions involving photosystem 1 and photosystem 2, we had different compounds like quinone, plastoquinone, plastocyanin, cytochrome F, cytochrome B6. All of these undergo oxidation reduction reactions. That is, each compound can simultaneously get reduced and oxidized when an electron is lost. Similarly, a compound can take up an electron and get reduced. And when two of the reactions occur together simultaneously, that is in continuity, A combines with B and A compound gets oxidized and B compound gets reduced. So, this is termed as a reductant and then there is an oxidant. So, in the process of oxidation reduction reactions, there is the flow of the electrons. Electrons flow. Now, this particular electron flow is coupled with the synthesis of ATP, that is adenosine diphosphate combines with inorganic phosphate to form ATP in the presence of electrons. It occurs in the presence of electrons. Whenever this reaction of ADP plus inorganic phosphate is linked to any of the biochemical processes, then you call that particular process wherein wherein ATP is getting formed, you call it as phosphorylation. Phosphorylation is the biochemical process whereby one of the byproducts is adenosine diphosphate combines with inorganic phosphate to form ATP. Now, this sort of a phosphorylation, which occurs in the presence of light, is termed as photophosphorylation. So, photophosphorylation is a characteristic feature of light reactions. Photophosphorylation can be of two types. It can be cyclic, when it involves all the compounds together, or it can be non-cyclic, when the electron is not returned. Either way, in cyclic photophosphorylation or in non-cyclic photophosphorylation, always there is a synthesis of an ATP molecule, which is an energy molecule, and it has got in its terminal phosphate, it has got the energy, and when that particular energy is broken, the energy is given for the sort of a cell, primarily in the dark reactions. Now, imagine a situation wherein we have phosphoglyceraldehyde PGA, it combines with NADPH, and primarily it gives rise to glyceraldehyde phosphate and NADP. This reaction is its NADPH2 2+, and this reaction is termed as a reduction reaction. Similarly, when you take the same sort of thing, what really happens here is you can have NADPH, which in turn can break down to form NADP. It can break down to form NADP plus H plus liberation of 2 electrons. This is termed as oxidation. If you take only phosphoglyceraldehyde PGA or PGL, phosphoglyceraldehyde, to this you add 2 electrons, and again it combines with the hydrogen, and it results in the formation of GAP, glyceraldehyde phosphate. This process is termed as reduction. So, these are common equations which are asked in MCQs. So, here we understand oxidation and reduction is coupled or joined. These are examples of redox reactions. Redox reactions are those which occur both in photosynthesis as well as in respiration in the chloroplast as well as in mitochondria, wherein a compound can get simultaneously oxidized as well as reduced. So, when there is a redox reaction, there is transfer of protons. There is always a transfer of protons. Now, the transfer of protons balances the movement of the electrons. Thereby, the particular sort of a compound remains as electroneutral. Now, this electroneutrality is very much important for a membrane. Otherwise, the membrane can suffer from bleaching. Thereby, now what we primarily understand is, if you take the processes of light reactions or dark reactions, they are very much dependent on the pH. The hydrogen ion concentration determines the pH. So, the pH of the stroma, the grana in the case of the chloroplast, or if you take the cristae and the matrix in the case of the mitochondria, are very, very important to drive light reactions, dark reactions, as well as you take the Krebs cycle and the electron transport system in the case of the mitochondria. So, the word here is coupled reactions are important, and all the reactions are redox reactions. And in redox reactions, first important property is there is transfer of protons, or the H plus ions. This balances the liberation of the electrons. Thereby the membrane becomes electroneutral. We call mitochondria and chloroplast as being pH sensitive because it reacts to H plus ions. So what do you exactly understand by the term redox potential? Any compound which is a component of the light reactions of photosynthesis or which is an integral component of the electron transport system in mitochondria, which occurs in the mitochondrial F1 particles. This any compound which has got the tendency to accept an electron. It accepts an electron from a donor. So you have a donor. Donor gives the electron to the substance. It gets an electron from a donor, and this same substance can donate the electron to an acceptor. It means a substance can take an electron from a donor and liberate and give the electron to an acceptor. So if electrons are taken and given simultaneously, it is taken and given simultaneously during an electron transport. It has the capability of oxidizing and reducing, and thereby you say the compound has got redox potential. So when you look at the light reactions, in the light reactions of photosynthesis, we understand according to the redox potentials, the molecules will be moving across in the membranes. And even in the Z scheme, of Hill and Bendel, the different components like plastoquinone, plastocyanin, they are arranged according to their redox potentials. They are arranged according to the sort of redox potentials. Now, when we talk primarily about energy transduction, how does energy transduction occur in organelles? That is, such as chloroplast as well as mitochondria in plants. Now, when you take the membranes, if you think this is the outer chloroplast envelope, and you think this is the inner chloroplast membrane, inner chloroplast envelope. These membranes are impermeable to H plus ions. They are impervious to H plus ions. So we have different sort of electron carriers. Electron carriers which are arranged asymmetrically across the membrane. When you take the membrane, the electron carriers are arranged asymmetrically. That is, it could be over here, so all of these, they could be quinones, they could be plastoquinones, plastocyanins, or they could be cytochrome F, otherwise it could be cytochrome B6. So when you take the electron carriers, these electron carriers they again reorganize according to their redox potential. When an electron is available, immediately the quinone will go and quench. It will go and remove the sort of an electron. So when it quenches, it gets an escape velocity, it goes up the membrane. So the electron carriers, when they are arranged according to the redox potentials, they are helpful with relation to transfer of electrons. We normally have the photolysis of water here in a membrane, due to which we have the release of the electrons, and electrons go across the series of electron carriers. So the carriers, when the carriers they take the sort of an electrons, this flow is termed as vectorial electron flow. It has got a direction. So it moves along the direction, taking the electrons from the inner chloroplast envelope to the outer chloroplast envelope. So normally this flow of electrons is against a gradient of protons. When you take the membrane, on the membrane we have an enzyme called CF1 ATPase. This CF1 ATPase is a special enzyme called coupling factor 1 ATPase. Now the coupling factor 1 ATPase, what it could primarily do is, it can translocate protons. That is, it can take up H plus, which is present on the outside of the chloroplast, and it can drop through its sort of a lumen, and the H plus ions will come on the inside of the chloroplast. So it builds up the movement of the protons, which is just like a sort of fountain, is primarily termed as a sort of a proton gradient. So we have a proton gradient which is coupled by CF1 ATPase. We also have a vectorial electron flow, which is because of the presence of the redox potential. So the proton gradient moves across downhill in terms of the electron carrier. So it moves always, electron flow is against the proton gradient. So concept number 1 is electron flow is against the proton gradient. While electron flow is always up, the proton gradient is always down. So electron flow is against the proton gradient. So during photosynthesis, during the light reactions of photosynthesis, there is the coupling of the electron flow with the proton gradient on the other side, which is attributable due to the presence of CF1 ATPase. Now what is CF1 ATPase? CF1 ATPase is a special enzyme. Now what does it primarily do? It helps in chemiosmosis. What is the meaning of chemiosmosis? In the presence of chemical compounds, if there is movement of protons through a semi-permeable membrane, thereby only a few of the ions can move in, then it is termed as chemiosmosis. So due to chemiosmosis hypothesis as well as electron flow, primarily the light reactions they are just driven. So CF1 ATPase acts like a sort of an engine, or it acts just like a sort of a motor turbine. So the CF1 ATPase keeps on spinning. So when it spins, the hydrogen ions, the protons keep on trickling into the sort of a chloroplast. So this is the basis of energy transduction. When we say we want to study up primarily with relation to light and dark reactions of photosynthesis as well as Krebs cycle and electron transport system in the case of mitochondria, the term it is linked to bioenergetics, because bioenergetics perceives the energy concept linked to molecules in biological systems. Now when you take chloroplast and mitochondria, both chloroplast and mitochondria are compartmentalized. When you look at the ultrastructure of the chloroplast, The ultrastructure of the chloroplast consists of the chloroplast membrane, the outer chloroplast membrane, the inner chloroplast membrane. Then it has got the stroma. Then it has got a pack which is called the grana. The grana is made up of a set of coins, just like a set of coins. The grana is made up of units which are called as thylakoids. Thylakoids are spirals, and many thylakoids can also get connected, and the connected thylakoids are termed as interthylakoid structures within the stroma. And again, when you take a chloroplast, the chloroplast also has got the DNA. Now, there is primarily the most important thing in the case of bioenergetics is there is compartmentalization. That is, the chloroplast and the mitochondria are segregated into parts. When you take the chloroplast, it has got a grana, it has stroma, it has thylakoids. Then the basic unit is termed as a quantosome. Then you have even stroma thylakoids. We also have the chloroplast DNA. These are all components of the chloroplast. When you take the mitochondria, the mitochondria has two membranes, outer and inner membranes. Then it has got the cisternae. The infoldings are called cisternae. Within the cisternae, you got F1 particles. The F1 particles are called as Fernandez-Morane particles. Then you got the lumen or the matrix. Parallel to this, we also have the mitochondrial DNA. So, when you take chloroplasts or mitochondria, they are initially compartmentalized. The reason why they are compartmentalized is because ATP can be liberated or synthesized by the process of chemiosmosis in installments. ATP can be liberated in doses, and again all the reactions are coupled. The meaning of the term coupled is oxidation reduction reactions, and in both chloroplast and mitochondria we have a proton gradient. The proton gradient is always energy rich or ATP rich. So, here we have, like, if you take the inner membrane of the mitochondria, or we take the thylakoids in the case of the chloroplast, they are involved in chemiosmosis and ATP. They show coupled reactions and they have a sort of a proton gradient. So, here primarily when you take bioenergetics in terms of photosynthesis, we understand there is generation of free energy from the electron transport system. In the form of ATP. Now, this energy in the form of ATP is used to establish, so that is, we have an electron flow. The electron flow and ATP is used to establish a proton gradient. How is the proton gradient established? It is proton gradient established by the involvement of CF1 ATPase. Where is the CF1 ATPase? CF1 ATPase is an enzyme. It is located between the outer chloroplast envelope and the inner chloroplast envelope. This is the outer chloroplast envelope and this is the inner chloroplast envelope, and we have the CF1 ATPase over here. Now what does the CF1 ATPase do? It primarily helps in channelizing the protons. Protons are channelized, and thereby it develops a proton motive force, PMF. It develops a proton motive force. So the protons, here we understand that the protons run through a transmembrane enzyme. This is called a transmembrane enzyme. CF1 ATPase is called a transmembrane enzyme because it exists between the outer chloroplast envelope and the inner chloroplast envelope. And thereby, when this keeps on going, when there is a churning, when there is a proton gradient going down, when there is an electron flow going up, in the presence of this, there is the synthesis of ATP. So thereby, the synthesis of ATP is coupled to oxidation reduction reactions, that is the electron flow, as well as the ionic flow through the membranes, that is when you say this sort of protons. So this concept is a very important concept in terms of bioenergetics of photosynthesis.